Aloha. Thank you, Anthony. Well, it's really my pleasure, my honor to be here. Very lucky. Hawaii is gorgeous, and you have some fascinating fungi, and couldn't be happier, especially with some of the colleagues I'm working with here as well. Anthony's a dream colleague. But I won't talk about either my specialty in Russ or my specialty in yeast today, but rather a couple of side projects that came out of a really long-term study that I've been doing in Guyana for over 20 years, and just some mystery fungi there, which are kind of fun to talk about, something a little outside of my usual expertise. But this, for any non-mycologist in the room, we sort of like to start with, people are always asking, why do you study fungi, right? What's the importance? And this group out of Germany, really, Mark Stadler's group, put some dollar signs just on the value of fungi. And they came up with fungi produce about $54 billion or trillion dollars of wealth a year around the world, and that's just doing ecosystem services. So there's some things like fermentation and drug exploration in here, but a lot of it is ecosystem services, which are really hard to estimate anyway. But you guys are all familiar with mycorrhizae, right? So most of our plants are mycorrhizal in some form or another. And this is, of course, a very important service that fungi play. But this kind of valuation assumes that we actually know what fungi are doing in the environment, right? That we know what they are and what they're doing in order to put dollar signs on them. And of course, that's not true for most of our fungi. When we even look at distribution of species richness on the planet, just very recently, in blue here is animals. I mean, very, very recently, we thought animals were most of the species richness on the planet. And it's only been in the past 5, 10 years that we've come to recognize that it's microbes that rule the world. Plants and animals are a very, very small slice of the estimated diversity of species out there. Most of this is microbial. And again, the question is, what are these species actually doing? How do they interact with other species? Now, the way we estimate fungi has taken a lot of turns through the years as well. And this is a paper that was in Nature probably 10 years ago now, but it looks at our three major eukaryotic groups. And what you're seeing there in green is the number of described species for each of these groups. And in gray is sort of the estimate for undescribed. So you can see immediately that plants, we have most of the estimated plant diversity described. Animals, there's a big range. It depends on what estimators you use. But here's the gray area for animals. And what stands out, of course, is that fungi, we have the smallest number of described species for these three big eukaryotic or multicellular eukaryotic groups. A lot of uncertainty, but a really huge proportion of undescribed species here. And again, this depends on what you use, how you do these calculations, and they can be all over the place. For instance, the very first paper that I'm aware of that really tried to tackle what this gray area here for fungi is a work by David Hawksworth in the UK. So he had an island system where they have a long history of natural history collections and surveys, and they knew basically how many plants they had and a good idea of at least how many macrofungi, that's the fungi that you can visibly see fruiting. And when he tallied up all the species, he came up with a ratio of 6 species of fungi for every species of plant in the British Isle. And since we have a good idea of how many plants we have, you can extrapolate that all over the world. There's roughly, at the time he did this, 250,000 species of plants. Then that 6-to-1 ratio would give us 1.5 million species of fungi. He's updated that a little bit to around 3.8 million in recent years estimated fungi. But there are other ways of doing this as well. There's looking at environmental sequence data. And so Heath O'Brien, also in the UK, looked at environmental sequences around the world and sort of estimated that we probably had about 5 million fungi, species of fungi, just looking at sequences coming out of soils. So this is soils only. So again, you can see that these might have some flaws with the Hawksworth. Does species richness stay constant, or at least in proportion to plants around the globe, or is it more diverse somewhere else? And we're just talking about macrofungi here. We're just talking about signatures of sequences. A later paper came out that estimates up to 156, or sorry, 65 million species of fungi. And this is based on looking at fungi that just associate with animals or just with plants, and then trying to extrapolate outward from that. My own work in Guyana, I used an estimating system that's pretty much the same as Irwin. Irwin's paper is the one that came up with this estimate for insects of 11 million. And so I used the same methodology he did from 20 years of sampling in Guyana and come up with 110 million, which again is probably, we know the Irwin now is an overestimate. This is probably an overestimate, but it gives you an idea that this really depends on what methodology you use. So the whole point is that our gray area, we don't even know how big or how small it is. If we go with the Guyana estimate, it looks like that, right? So there's a lot of fungi. And again, to me, it's not just finding these species, but every species has a story. These are unique species. They're adapted for their habitats. They're interacting with the environment. So what are they doing? Excuse me. And so I'm going to take you to Guyana, where we'll just look at a couple of these that we've sort of worked out the story for them. So if you're not familiar, it's up here in the northeast corner of South America. And this is a region called the Guiana Shield, which is this it's the oldest geological formation on the planet. And it's all sandstone. And through times, Guyana means many waters. And through times, all these riverine systems have eroded parts of that sandstone so that you're left with these big Tepui mountains. This is Mount Aingana back here. But this is the region where Conan Doyle wrote The Lost World, or if you've seen the movie Up, these take place up on these tepuis. They're very remote and difficult to get to. But part of the beauty of working up here in Guyana is it is very remote, but it is a pristine forest. And so we can really get a good handle on what things look like before there's human disturbance. There's a lot of other really cool ecological reasons that we work in this region. But anyway, that's the important take home for this talk. So basically, we sampled out here using a plot-based system for about 19 years in the Pacoima Mountains. And then since then have also done sampling in other regions of Guyana. But to get out there, you gotta get on a little Cessna. These things can hold 600 pounds. That includes your body weight. So you can imagine you need to really limit the amount of gear you can take out there, miniaturize it. You're also packing your rations and things out there with you. When you land, well, here we are flying. This is the Pataro River. Almost the entire population of Guyana lives on the coast. There's almost nobody in the interior except some indigenous groups. So it's pretty much all pristine. But here we are flying up the Pataro. This is Mount Aingana, one of those tabletop tepuis there. Fly over Kaitur Falls. This again is the Pataro River. Kaitur is the second tallest waterfall in the world. Anybody know what the tallest is? Could have been. It's Angel. It's in Venezuela, but it's also part of this Tepui system. So when you measure single drop. Anyway, this is our second tallest one. And again, we keep going up the Pataro here. And then we actually land in an old abandoned mining camp where there's enough clear space that we can land these Cessnas. And then these guys become indispensable. So these are members of the Patamona tribe. They're indigenous hunter-gatherers that work back along the Brazilian-Guyanese border, and basically been working with the same guys for over 20 years. We're all getting old and decrepit together, but we couldn't do anything without them. So they will hike in and meet us where the plane drops us. And then we load everything up. This is a dugout. So got to start portaging upriver and getting around the falls. So there's a lot of portaging. Hiking through, there is some savanna area that we can hike through. And lots and lots of water crossings with our gear. This is the one we call the Monkey Bridge, which we do not look forward to every year, trying to get all our gear across the Monkey Bridge there. And then we get to the place where we've more or less set up a permanent site. Obviously, shrubby, a lot of secondary growth here because we have cleared the area. We've got to rebuild the frames every year because they'll rot down. But build the frames and eventually throw some tarps over it. There's sago, making sure everything's nice and tight. Just use bush rope. These are lianas or the inner bark of some of the trees. It's nice and springy that we can use to tie everything up. And again, put the tarps over it. And then this is where we live. We built some little tables. And our sleep area back here is our hammocks, our kitchen area. And this is actually the research part of camp right here. And that is my desk that I rebuild every year out of palm. But that's where all the magic happens out here. Now, I have through time taken several students and postdocs out here, and I want to call out Rachel here. Rachel Cook did her PhD with me and then a postdoc, and she worked with me out here for 6 or 7 years. And a lot of the stories I'm going to talk about today are just side stories that Rachel and I did together. If you're wondering how we can light up our microscopes here, we're actually using our headlamps and sort of rigging it up so that we can do our microscopy at night. We're on the equator, so we only have 12 hours of daylight. It is dark at 6, so if you want to keep working, you got to rig a lot of stuff up. And so the first story is this thing right here. So I began noticing this white looking stuff dangling from trees at one point, made some collections of it. It's clearly a fungus, but no idea what it is. No fruiting structures, no spores or anything. Brought it back home, did a little research, and it had been described from Suriname, but about 100 years ago, but very little known about it. And what's interesting, so sometimes you can find it just dangling from trees like this, but often it's part of these litter traps. You guys know what litter traps are? So a lot of times in these wet forests, the turnover is incredibly fast. And some of these fungi will form rhizomorphs up in the canopy. And when litter falls from the tree, it gets caught in that. It never even makes it to the ground. It's like big webs to trap the litter, and it will decompose up there. And so eventually some of these things will fall down. So here's one that fell down on the ground. You guys know what rhizomorphs are? I see some, yes. So that's a fungal vegetative structure, but it's this ropey stuff. And they can be different colors, white or black, but obviously this guy forms a white one. Very easy to recognize, forming these litter traps that fall down. So there's not much going on with this fungus, though. Here's a little bit of that trap. We've got the white rhizomorphs and some of the leaves. They form these structures on the leaves as they fall into the traps called a byssi. And that's basically where it attaches to the leaf and gets the nutrients out of it. But as far as morphology, there's really not much. When they get more mature, they put up these little knobs all over it. But if you look or take a cross section, there's not much going on. There's a core and an outer surface to these things. Again, no spores at all, no type of fructification on these. The outside is full of these, what we call generative hyphae, which means they're very flexible. And they have these things called dendrohyphidia, all these little curvy branches on the end of them. And then the inside is just skeletal hyphae. That's really hard hyphae, long with thick walls. It gives it a lot of strength. So they've got the strength and the flexibility, but nothing else. As I said, nothing else that you could use to identify the structure. We did find that it had been described from Suriname as something called Rhizomorpha corinicarpus. And Rhizomorpha is one of these junk genera that we used to use for vegetative structures. So it doesn't give you any information at all about what kind of fungus, what even phylum it belongs to. It's just a name for a vegetative fungus. So the first thing we wanted to do is figure out, where does this actually belong on the fungal tree of life? And so Rachel and I sequenced a bunch of them, and it actually belongs in a mushroom family. So this is the Marasmiaceae, and Marasmiaceae have a lot of beautiful mushrooms. Marasmius is very common. These are common litter decomposers, especially in wet tropical environments. So you'll find them on leaves in the litter all the time. Things like Crinipellus and Campanella. You get some little reduced things. This is a Cheocolathus. It's about 1 millimeter in diameter. Just looks like a little cup, but it's actually a reduced agaric. This is an unusual fungus, Monilioptera rorii, that attacks cocoa pods. But it just produces conidia, but all that white stuff is conidia on the surface of the pod produced by the fungus. And the one down here at the bottom is Brunio corticium. And it's what we would call a corticioid fungus. So it doesn't form a mushroom, it forms a flat hymenium over the surface of whatever it's decomposing. But that hymenium is full of spores. And it turns out that that is the sister to our Rhizomorpha, this Brunio corticium. Here's what it looks like under the scope. Of course, it is sexual, so it's producing spores, but it does have these dendrohyphidia, you remember from our fungus as well. And so we were able to at least give this thing a good name, put it into Brunio corticium. But it raised another question. All the collections we had and the literature accounts of this thing, however sparse they are, there is no evidence of any fruiting at all, no spore formation. It's just purely vegetative. So you've got to wonder, how does this thing get dispersed if it's a vegetative fungus? It's up there in the canopy forming these litter traps. So does it get dispersed? Is this something that's really, really endemic? Dispersal, you know, is really important for a lot of reasons, but it can also give you a hint of how things are adapted for their environment. So the first thing we did then is start looking for the fungus elsewhere. So we knew it was in Suriname from the first reports. We knew we had it in the Pachyramas. So I called up a really fantastic neotropical mycologist, Jean Lodge, who worked out of Puerto Rico, did a lot of work up in Central America and the Caribbean. And her husband is an ornithologist who's also a good mycologist as well. And then we had another friend in Brazil, Suzanne Sorel. And we started looking for this thing, and we found it everywhere. So didn't find it in the Caribbean, but we found it in Brazil, through most of South America, at least the tropical part, and all the way up to Belize. So this isn't an endemic fungus. And again, none of the collections that we were getting in had any hint of any kind of sexual structure on it, never been observed. But when we were looking for it, we started finding it everywhere in birds' nests. So birds are incorporating this in many of the different countries, and many different species of birds seem to be incorporating this as nesting material. And this guy over here, this bare-necked fruit crow, I got curious about him because you see he's actually got a piece of a trap here in this photo. Looked him up, and here's the picture of him from eBird. And he's actually carrying this nesting material around in his beak. The range of this particular bird looks extremely, very much like the range of our fungus in South America. So our hypothesis is that this is a fungus. It's adapted as a litter decomposer that forms these big webs. It gets picked up by birds as nesting material, and then it grows out from there. It's a nice elevated position to continue growing out in its webbing behavior. But while we were looking at birds' nests, we started noticing, of course, that birds are using a lot of fungal material in their nests, not just the Bruneo corticium. But here's a bird nest that looks to the untrained eye, or to my eye, like it's full of roots or something. But when you start looking down, these are rhizomorphs that it's using. Here's the little mushroom fruiting right from it, from an abandoned nest. So we got curious about this phenomenon as well, and looking at rhizomorph usage in general in birds' nests. As far as, so it's not reported, or wasn't at the time really reported in mycological literature, but there were reports of it in the ornithological literature, this use of rhizomorphs in nest construction, but almost always attributed to the genus Marasmius, if it was identified at all, and almost always to a single species called Marasmius kriniseki. So remember, Marasmius is related also to our Bruneo corticium. So birds will use nesting materials for 3 different parts of construction. They'll use it for attachment, so just to secure the nest to the substrate. They'll use it for structure, that's outside, that holds everything together, obviously. And they'll use it for lining. So some birds you'll find nests that are just all rhizomorphs just on the lining. And again, almost always, if it's attributed to a species, it's attributed to the species Marasmius kriniseki. And so what Rachel and I did is spend one summer collecting abandoned bird nests and teasing out all the rhizomorphs, and using, of course, DNA analysis to see if these were Marasmius kriniseki or not. And so here are the results. The color is not really popping, but where you see the greenish blue, those are rhizomorphs that are coming from birds' nests. And just there, basically in our little tiny area of Guyana, we came up with 25 different species of Marasmius that were being incorporated as rhizomorphs in birds' nests. Not a single one of these is kriniseki. We have yet to find kriniseki in a bird's nest, but almost all of them are undescribed species. We were able to get 2 of them described. You have to have the mushrooms really to describe a Marasmius species, and since a lot of these we were getting just as rhizomorphs, we could only describe 2. But the other thing we did, we had kept notes of which part of the nest they came from. And it turns out that this is not, of course, statistically very robust, but just looking at the data, there does seem to be some sort of selection going on where some of these species are being used just for structure or attachment, at least within our data set. And some of the species are just used exclusively for lining. So there might be some selection going on. So our next question was, why would birds be maybe selecting these materials for nest construction or for different parts of nest construction? Well, this has been answered somewhat by an ornithologist in Brazil who looked at rhizomorphs being used for structure and attachment and found that they're much stronger and more hydrophobic than roots, for instance, than any other nesting materials birds use. So in a wet tropical environment, having something that's really robust and hydrophobic, you can see where that would be adaptive. So what about the lining? Nothing in the literature to suggest this, except that we know that there are lots of Marasmius species that produce secondary metabolites with antibiotic properties. And if you think about a bird in a wet tropical system, that parasite pathogen load is going to be pretty high for those nestlings. So if you can incorporate something that's producing antibiotics, you might be able to reduce the parasite load for the nestlings. So that's our hypothesis. To test this, the first step we had to do is see if these are even viable, these rhizomorphs inside the nest. So I realize you can't see this, but we would surface sterilize rhizomorphs, cut them up, do culturing with them on plates and look for growth. And here you can see the fungus growing out of the sides of the rhizomorph. So they're certainly viable. We've got lots of cultures of these now. And I have a graduate student who's characterizing the antibiotics that they're producing to test that hypothesis. So that's the to be continued part of the story with the rhizomorphs. So stay tuned for that if we can characterize those antibiotics. But I'm going to finish up with this one other really cool fungus that we did a lot of work with. This story actually begins in 2010 when my colleagues and I found this weird looking fungus. It's what we call subhypogea, so it's only partially buried in the ground. It looks like a truffle or something. In fact, my field notebooks say truffle when I first collected it. But when you dig down, it's actually always found on roots. So it's got this little remnant of a stipe, but it's on buried roots. And when you cut it open, this is very young and this is very mature. It goes through this maturation process and turns bright red at the end. And this thing, we described it as a new genus, Gyanogaster. This one, Gyanogaster necrorhizis, because we suspected it was a pathogen being always associated on Decimbe roots. But it was a bit of a mystery to me, because when we talk about what we call a sequestrate fungus, a fungus that produces the spores on the inside. So this material here is called a gleba, when it's a mushroom that produces spores inside, and this is full of spores, and this is the mature stage. And when this fungus is mature, this gleba is very, very gelatinous. So the thing is hard as a rock. I mean, you couldn't bite into that. It's really, really hard on the outside and gelatinous on the inside, where the spores are produced. And it just always puzzled me how it was being dispersed, because when we think about fungi, there are things we call sequestrate syndromes or gastromycetization syndromes in mycology, where mushroom-forming fungi will start to enclose, but they do that in very specific cases in response to a certain set of factors. So some, like your puffballs, everybody knows a puffball. These are wind and rain dispersed, and when rain hits the top, it'll send out a puff of spores. Those spores are inside. So when this thing's mature, the gleba, the stuff inside, it's very, very powdery and dry. It's not gelatinous. There's usually this exoperidium, the skin on the outside. It's very thin so that it can easily be cracked, or it might have a hole at the top for the spores to escape. And these are saprotrophs, as far as we know. And this is a syndrome that's occurred many times in the fungal tree of life, but they all tend to adopt the same sort of characters. And then we have those adapted for mammal dispersal. Again, they're going to have a thin exoperidium, because the mammal's got to be able to bite into that. They can be a little bit gelatinous on the inside when they're mature, but they give off odors. So think of truffles and things like that, your classic mammal dispersed sequestrate fungus. So some kind of olfactory cue when they're ripe, so that something will come dig them up. And this gleba is very, very soft, as is the exoperidium at maturity. And these are almost always ectomycorrhizal. And so they tend to be buried for the most part, whereas these are exposed, of course, on the surface. So our fungus, what was puzzling me, first it has this really hard exoperidium. It's hard to imagine a mammal chewing through that. It's subhypogeous, so it's neither epigeous or hypogeous, but it's somewhere in between. The gleba is very, very gelatinous and thick at maturity, and no odors that were detectable to us, at least, at any of the maturity stages. And we suspected it was a pathogen because it was on roots. So how's this thing get dispersed? Well, usually if you want to figure out how something gets dispersed, you can look at the population biology, look at how far it's being dispersed, because that'll give you a hint of what the vector might be. And so that's the approach we took. Now, the first thing we had to do is figure out where that guy resided on the fungus tree of life. And it turns out that it is a sequestrate relative of Armillaria. You guys know the genus Armillaria? So these are notorious plant and root pathogens, right? The Armillaria species. So that gives us, again, some hint that it probably is pathogenic since Armillarias are. Armillarias are also notorious for forming rhizomorphs for vegetative dispersal in addition to the mushrooms. Our Guyana forms rhizomorphs in culture. So this is actually it in culture, these non-melanized rhizomorphs, but we never saw it producing rhizomorphs in nature. But to get back to what might be vectoring it, if it is pathogen, we went out over 4 or 5 years and collected all the Guyana at different patches, got a genome and developed some SSR markers at the time, 11 polymorphic markers for this. We classified all of our Guyana from in what we call maturity groups, basically, but the immature, the young, the almost mature, and then the very mature. So we just classified them into 4 groups and did these analyses to see how far these genes might be flowing. Now, as a control, we also, to make sure our methodology was good, looked at another, what we call gastromycete out there in the system. This is a fungus. This was, I think, the first fungus we described from Guyana. This is Pseudotelosoma volvata. And when we described this thing, it actually had a lot of press for a fungus in the mycological literature. It was the fungus of the century. This was described in 2001, so the century was 1 year old, but nonetheless, everybody was really excited about it because it's related to penicillium, if you know penicillium, a little mold. And here we have this great big persistent woody fungus that is ectomycorrhizal with the cyme beet trees out there. It doesn't look anything like a penicillium. But at any rate, we do know that this one is wind and rain dispersed. So we use this as a control and also collected populations of this guy. So we did this work up at our Pitaro site, and we also went to another site, Mabora, which is about 100 kilometers away from Pitaro, where both fungi exist, and looked at gene flow. And first of all, as predicted, Pseudotelostoma showed lots of evidence of gene flow between the Pitaro and the Mabora sites at 100 kilometers. But nothing, not a shared allele with Guyana between Mabora and Pitaro at 100 kilometers, which is pretty astonishing, really. So we went back and looked at the data a little at a smaller, finer scale. So this is our base camp, that airstrip that I showed you where we land. That's that population there. Here's the river right through the middle. So we roughly classified our patches into 4 populations, 2 on this side of the Pitaro and 2 here, and then looked at the gene flow there. And basically what structure was telling us is we had 8 populations. If you're not used to reading these, everything over here in red have shared alleles. So the same color would be shared alleles. But there are no red alleles, for instance, over here. And we're looking at a distance of about 4 kilometers and no gene flow between them. We had actually a good deal of gene flow over here, but basically none from the airstrip or over there in Sussan. If we look at the number of multilocus genotypes, just the individual genotypes, almost every basidium was a unique genotype, a very high percentage. So it is clearly sexually outcrossing. As we suspected, these were basidiospores being produced. We don't have a clonal situation that would explain something like this. And when we did spatial structure, it was really telling us that the most that the basidiospores were dispersing was like 5 to 10 kilometers. Again, that's a really, really tiny distance for a fungus. So it's not wind and rain, and it's probably not a mammal. So what the heck is doing the dispersal for these guys? Well, on the mature ones, we would often crack open a mature one and they'd be chock full of termites. And you can see this guy tunneling all through there. So we began suspecting perhaps termites are actually doing the dispersal for this fungus. Here's a regular termite from the region, and here's one that's been feeding on Guyanic aster. And you can see it, they really do feed on this. It's got gleba just chock full in its guts when we pull them out of there. So they are eating this. And our first hypothesis was, well, this is going to be a gut passage syndrome. So there are fungi like truffles that have to pass through an animal gut in order to have viable spores out the end. So we looked at that. Here's spores from the gleba. Here's spores from the frass. Would make the poor termites poop for us. And the spores just are not viable. We couldn't get any of them to germinate, and they're all chewed up. So it wasn't gut passage. So what was going on? Well, you see spores here? Remember, these things are really gelatinous at maturity. They're forming all this goo, and the spores are just sticking to the outside as they're in their feeding of the exoskeleton. Yeah, just beautiful, even up under his mandibles here. But they've got these spores stuck all over them when they feed on the mature stuff, and then presumably wander off and disperse those spores to a nice woody substrate, which is their preferred habitat. So we thought maybe there was a coevolutionary story going on. Turns out there's not, at least not when we're catching it, because we found multiple different species of termites inside these things. They are all from the wood feeding group of termites. So they're not soil feeders, they're wood feeders. But there are many species that are attracted to Guyanic aster. And then, it made good sense, if you will, for the fungus to be adapted like this, because it's getting a nice targeted vector. But what's in it for the termites was our next question, right? Also the termite foraging range out there overlaps at least decently with the dispersal range we were looking at. And to test this hypothesis that the fungus was actually pathogenic, we did, of course, look at the genome, and it certainly got all of the right genes to have a pathogenic system in wood. It can, God, my English has left me now. It's capable of degrading cellulose and lignin, and this is actually roots with the mycelium inside it from the Guyanic aster. So why might the termites actually be doing this? What's in it for the termites? Well, when we tried to do tissue cultures, I showed you that beautiful culture forming rhizomorphs. There is a technique that mycologists, especially people who work in mushrooms, depend on, and that is that the inside of a fruiting body is going to be sterile. So even if you've got a thick mushroom cap, if you just crack it open, take tissue outside, you should be able to get a tissue culture because it's sterile. And so even with our younger Guyanic aster, if we carefully crack that open, take tissue out of the inside where it should be sterile, we were getting tons of bacteria instead of fungal hyphae all over the place, just chock full of bacteria in there where we shouldn't have so many bacteria. And so at first I would get frustrated and throw the plates away. And then Rachel, who's much more clever than I, said, well, let's find out what those bacteria are. And so she started sequencing all these different cultures we were getting out of Guyanic aster. And they all turn out to belong to the Enterobacteriaceae, everything we could get, or the proteobacteria, but in the enteroids. And here, for instance, is a lot of our Guyanic aster bacteria. And so we did some studies to see if these came from the soil or the wood or something else. So we did some microbial analyses of the different populations. Again, these are different maturity groups. And then here's the soil. And really the take-home message is that the bacteria inside the Guyanic aster are quite different from those inside the soils. And in fact, sorry, I'm having trouble seeing from over here, but anyway, the mature is really different from the other classes as well. So we've got some sort of maturation or selection going on just within these basidiocarps. The main component in the mature ones is this guy here that looks like a new genus. We're not good bacteriologists, but anyway, it's not anything that's been described. But the groups that it falls into, these bacteria play really important roles in the environment. The most important, maybe, hint for us was that they're nitrogen fixers. And anybody who works with wood decomposers or anything that eats wood knows that wood is really limited in nitrogen. Termites are nitrogen starved. They are always looking for extra sources of nitrogen. And so our hypothesis at this point in time was maybe that's the reward. Guyana is offering a nice nutritional supplement for these organisms that are starved for it. So we went back again and we did 2 different assays, dug up a whole bunch more Guyana and looked both at the percent nitrogen inside the basidiocarps and at the activity of nitrogenase. That's the enzyme that the bacteria use to actually fix nitrogen. And we do see this is compared to the roots that we dug them up off of, that there's a lot more nitrogen inside, especially the mature Guyana, but also a lot more nitrogenase activity. So it does appear this enzyme is not being produced by the fungus, that's being produced by the bacteria, and they are actively fixing nitrogen inside these basidiocarps. One adaptation that the fungus does have to help this process is it's doubled the number of superoxide dismutases in the genome. And this is also upregulated, especially in the most mature sporocarps. But that's an enzyme that helps keep an anoxic environment for nitrogen fixation to occur. So we looked at how, because fixing nitrogen is very energy intensive. So we looked at how that energy might be generated. And it does look as if the Guyana is doing fermentation inside to produce the ATP. It certainly got all the right machinery for it. And in our most mature Guyana the products are there as well for fermentation. We looked even at why it's so gooey, because this is something that seems to be unique to this sequestrate fungus, but it does have this cetalizin-like protein produced by Guyana. That's a mucilage protein like Arabidopsis seeds and things like that have it. So we suspect that's what it's producing to stick in the mature gleba, the spores to stick. And one of the really more exciting findings, this entire secondary metabolite cluster here is all upregulated just in the actual mature Guyana, not the other ones. And it includes 2 terpene synthases, which are probably, we hypothesize, is signaling for the termites. Those are basically the signal that we're mature, your nitrogen supper is ready. Yeah. They're the second and third most abundant proteins in the mature gleba are these terpene synthases. So this is our hectic little model for what we think is going on here. But basically, as it's got several species of bacteria in it, but as they mature, it's just the nitrogen fixers that are really going strong. It's producing more nitrogen that's getting sequestered inside this basidium, and then at maturity, it's releasing the terpene synthases, and then of course the termites come to feed, and the spores adhere with that nice mucilage, and then it carries them off to a new substrate, which is really, really satisfying. I don't know how many times in my career I've been really satisfied that something that puzzled me morphologically, like why does it have such a hard exoperidium? Well, we think it's to help keep an anoxic environment, and it doesn't bother a termite mandible to chew through that thing? Or why is it so gelatinous at maturity? Well, it helps the spores adhere. So all these things that were really, really puzzling about the morphology of this thing kind of make sense within this model. And as far as we know, this is the first example of nitrogen fixing associations in a fruiting structure, not within the hyphae or the vegetative structures. Certainly the first instance we know of termites selectively feeding on mature fruiting bodies. They do feed on hyphae in Africa. There's a Termitomyces hyphae that they farm, but this is the first case we know of this kind of behavior in the mature fruiting bodies, or even of arthropod dispersed sequestrate fungi. And it just highlights again that fungi, if you don't already know it, they have very flexible metabolisms, and it allows them to adapt to lots of different situations and organismal associations. Yeah, so thanks to especially the Patamona, my crew. Here we are waiting for the plane to come pick us up again, and to Rachel working hard. And thank you. I'll take questions.